Very good morning. I'm Dr. Raghavamma, presenting on culture presentation methods. These methods are widely used for preserving the microbial cultures for different purposes. The methods can be grouped as two methods. One is a continuous metabolic culture methods and suspended metabolic culture methods. And under continuous metabolic culture methods, we can talk about the periodic transferring, overlaying with the mineral oils, and making a saline suspensions. And under metabolic state cultures, suspended cultures, we can talk about the storage in a sterile soil, drying with a dry vacuum, lyophilization, use of liquid nitrogen, and storage in silica gel. While coming to the method of periodic transfer, which is widely used in small scale laboratories mm, or research organizations or for purpose of laboratory experiments, in the organisms will be periodically will be transferred into a fresh medium, and after growing the organism for about 25 hours at the respective incubation period, the organisms will be further preserved in a refrigerated condition for over 1 month, and periodically the organism has to be transferred into a fresh media for every month. The major drawback of this technique is like this: the culture can be preserved only for about 3 years, and after that period, the organism undergoes mutation. Overlaying the medium with the mineral oil. So, this is a method which is very simple and easy to perform, which has a similar drawback as that of the periodic transfer method, but the organisms can be taken directly from a medium after incubation period without discarding the mother cultures. The organisms also can be kept at the normal laboratory temperatures, and refrigerated conditions are not required. Using saline suspensions is a method which is mostly preferred for transferring the cultures from one place to another, where the metabolic activity of the organism will not be undergone. Only the moisture conditions will be maintained in order to maintain the organism in its normal state. Then the other type of methods where we completely dry the organism is storage in a sterile soil, and this method is widely used for storing the spore form of the organism, mostly in case of fungal cultures. And lyophilization or freeze drying method is one of the widely used method for preserving the cultures in a larger volume and for an infinite period of time at room temperature or normal laboratory conditions, in which the organisms will be placed in a vial subjected for drying at minus 70 degrees centigrade, and further the devacuumed so that whatever the moisture content of the cell will be sublimed, and the total culture will be in a powdery state, a dry powder form. So this vial after sealing is can be placed at normal temperature conditions, but the major drawback of this method is once the vial is open, it has to be subcultured and transferred into a normal nutrient medium, and the organisms may undergo some sort of destruction due to freezing and drying. The other method which applies a freezing technique is use of liquid nitrogen method, but here as the organisms are being placed along with the cryoprotectants like dimethyl sulfoxides, the bursting of the cell can be prevented, And the cultures will be first dipped in liquid nitrogen, that is at minus 196 degrees centigrade, and then they are preserved over the liquid nitrogen. And you can also store the cultures for an infinite period of time in this condition. Storage in silica gel is similar to that of storing in a sterile soil, which are mostly used for the type of spores. Okay, thank you.